mechanisms and scientific questions. And obviously we're not physicians and we can't answer all your questions, but it's really an opportunity if you have anything that you would like to ask these folks to get some better understanding about the drugs that they are, the compounds they are developing at the moment. So before we start, what I'm going to do is I'm going to pass the mic to everybody so they can just introduce themselves to you and tell them they can tell you a little bit about what it is that they do. I'll start over here.
Hello, my name is Dietrich Haubenberger. I am from Neurocrine. It's a company that I think was introduced to you yesterday. Brand new. The company is not brand new. Our company is around since the early 90s. We're a company based out in San Diego, California, so excuse me if my body is still on the West Coast time. I came up here for my first meeting at 7 in the morning. So we are partnering with Xenon, and that's really big news also this week. And we're partnering for the development of Xenon 901, which is the very selective 1.6 channel inhibitor. And I'm a neurologist, so anything regarding to basic science and mechanism, I really can't answer. This is why you have the Xenon colleagues here. The only thing that I can say is that I'm incredibly excited and awed by being here, and actually I would like to turn this around and ask you so many questions, because we having this opportunity to come to you and to partner with Xenon for the development of a hopefully new drug is very moving to us. We have as company quite some experience developing drugs all the way from phase 1 to phase 2 to phase 3, all the way to market approval. We have 2 drugs on the market that treat. One of them treats a hyperkinetic movement disorder, valbenazine, and the second one that is endocrine hormone-based treatment of endometriosis. So we're excited to be here. We want to learn a lot as we go together, through these next steps of development. And I'm going to hand it over.
Thank you. My name is Alison Cutts. I have a PhD in genetics, and I've been with Xenon for almost 20 years now, so I've been around, I guess, even longer than some of our projects, including 901. So I'm happy to answer any questions you have, around human genetics, molecular genetics, preclinical drug development, and maybe I'll leave the mechanistic aspects to Praxis. But thanks for the opportunity to be here. It's a real honor to be part of this gathering, and feel free to ask us questions. I can't guarantee that we'll be able to answer them all, so I will preface this by saying, sometimes we don't know the answer ourselves. Other times, we may be having internal discussions, but we're a publicly traded company, and we can't always divulge some details, but we will be as transparent as we possibly can. But I just ask for your understanding in those regards.
Thank you for saying that. So my name is Marion Whitman. I work with Praxis. We're the new kid on the block. We've pretty much been around for 3 years. We have been very close to this community. We've been seeing this community grow while we have been growing, which is actually very exciting. Our very first project was very much focused on SCN8A epileptic encephalopathy, and I spoke about that yesterday. It's Prax 330. My role at Praxis is I'm head of biology as well as toxicology, and so I'm very happy to answer any questions about the preclinical process of developing drugs for your children. And I think that's enough for me.
Hi, I'm Celine Grayson. I'm from Xenon. I've been at Xenon for almost 10 years now. So, similar to Alison, I'm a molecular biologist by training, but my role at Xenon for the past several years has been in clinical science. So I work on our paediatric epilepsy programs and our planning for the clinical development of our compounds. And I was the person who was kind of behind the recent survey that we conducted in collaboration with the Acute Syndrome Foundation. So I'm the person who's going through all of that data right now. So I just wanted, on a personal level, just to thank anyone who participates. I'm sure there are several of you in the room and we really, really appreciate it. We know that you're asked to share this information time and time again, but it really is so valuable to myself and my colleagues as we're trying to plan clinical trials, to just have that information in our minds, and to hear your personal experiences, just rather than the information that we get from the literature. So really, thank you very much, and it's a great pleasure to be here.
And I'm Chris Cayley, so I'm also, I work with Marion at Praxis. I've been at Praxis for about 2.5 years, almost 3 years. But I've been in this field, sodium channels, looking at mutations since 2004. So Dravet type mutations, then moved to SCN2A, and now, obviously this SCN8A, now 6, but I've worked on pretty much all of the channels, including the cardiac channel, and so I'm very much the basic mechanism person. So I would be in the, when you get the genetic change, we make the mutation, try to figure out what's going on, and then how can we try to reverse that by physical change to restore normal functioning to the channel. So that's my role at Praxis. I think that's sufficient. And yeah, even at the basic science level, I concur with some of my colleagues here. We've been doing this a while, but we still don't know everything about the basic science, but we're definitely working and happy to share what we do.
Thank you guys. You can take a seat then, and you can so I'm going to walk around with the mic as folks have questions. So, do you have a question? Okay, great. We'll start.
The two companies articulate how the compound that each of you are developing differs in the way it works in the mechanism in the channel.
So maybe Chris can start with Praxis.
Absolutely. I think that's a great question, something to keep in mind. So they're completely different types of mechanisms, completely different types of molecules, and they're both really exciting. So the Praxis molecule would be more of a traditional sodium channel blocker. By that, it has the ability to block all of the isoforms, but it does so in a very unique way, very differentiated than the current molecules like penicillin or carbamazepine. What we heard from Marion yesterday, that means it's able to basically, what we believe is able to block the abnormal activity of the channel, but leave all of the normal functioning intact. And that's for all of the isoforms. So unless there's some pathology, we don't think we're really at the concentrations we need, we're really not blocking normal activity. The xenon molecule binds a different part of the channel, and this is some of the newer molecules that have been in the pain space and other fields. That other different binding site actually gives you very exquisite isoform selectivity. So it's a very selective molecule for NaV1.6. But they differentiate, even in binding kinetics, they differentiate in how they interact with the neuron. Both mechanisms are plausibly efficacious, and they may be good for different types of patients, good for different types of mutations. So I can't confidently say one would be better than the other, but I can confidently say they're very different. So it's exciting to have two shots on goal in this population.
Sure, I would agree with you that, mechanistically, they do behave differently in cell-based models, in animal-based models. One of the ways that we will differentiate these compounds is in the clinic, hopefully. And I think you're right, is that, some compounds may be more efficacious for certain mutations, certain individuals, and other compounds, for a different subpopulation of AD-related epilepsy. I guess the only thing I would add to you, just to make it clear, is that, or clearer, is that XEN901 only inhibits the NaV1.6 protein. It doesn't inhibit any of the other sodium channels. And so, in that respect, it's a, excuse me, a precision medicine approach for your kids.
Thank you. Anybody else have a question? Follow-up question?
This is more for the Praxis group. Obviously, with 901, you guys are already starting your phases. With your drug, you're not on human trials or even starting phase 1, correct?
Yeah, so Prax 330 has been in adult healthy volunteers. That study was conducted last year, and we're still evaluating that data. As far as we can tell, as data is coming out, we'll be able to share more on that early in 2020. So it is in clinical development, but Senai behind where 901 is right now.
Question overall, with the new drugs that are coming out, are there going to be age or weight restrictions that's not going to allow someone like my daughter, who's actually going to be 2, but she's only 14 pounds, is she going to be able to take these drugs, or are they just going to be out of her weight range?
Alice, will you be able to?
Well, I can't give a complete answer, but I and my clinical colleagues will comment as well. What I can say is that certainly at Xenon we've put a lot of thought into being able to dose young children, and so really that's twofold. One of them is a regulatory requirement to do what's called juvenile toxicology, so you do some toxicology studies in younger animals to try to better predict how the drug would be tolerated in a pediatric population. And the other is specifically around generating a pediatric formulation. So when we did our adult healthy phase 1 trial, we had a tablet that adults could swallow. However, we know from our interactions with all of you and from the literature that we needed to have a pediatric specific formulation that was compatible with many of the technologies that you use every day, including feeding tubes, that was palatable, that could be mixed with liquids. And so we have developed that formulation, and we're currently testing that formulation, not in kids yet, but in healthy adults, and in part because we want to show that the pediatric formulation behaves like the tablet formulation behaves in adults, and that will just give us additional confidence around what's called the pharmacokinetics, which is, how high did blood levels get, how long does the drug stay in the system, things like that. So that's a second phase 1 study that's currently ongoing to compare the pediatric formulation to the adult formulation that we've already tested in the first phase 1 study.
Maybe I can add to that. It is certainly the goal that a drug like this will become available to a patient population that can benefit the most of it in a safe way, and to learn about that. That's basically the clinical development, right? To learn about the safety of it, learn about the efficacy. So. In a clinical development program, we look exactly at that. Does the drug behave differently in different weight groups? Does the drug behave differently depending on different other medication that might be there? Does the drug behave differently on certain symptoms of it? That's all now the learning phase that we're now into. But again, the goal is really to demonstrate efficacy and safety in a population that can benefit of it the most.
I guess to more specifically answer your question, we are working with Neurocrine. They're a new partner with us to design this trial, and so we don't have specific inclusion or exclusion criteria at this time. Those will be developed and they will be part of the clinical protocol, but we are not able to share that currently.
Thank you.
I can just say that, we're very much in the same boat. Your first clinical trial needs to be very carefully designed, and your enrollment criteria have to be very clear, and so it's hard to say how it'll pan out, but the medical directors are thinking very, very carefully about how to design that trial, keeping in mind that also the overall patient number is very small, and so there's a balance between running a good trial to understand whether the drug actually works, but also not excluding everyone so you can still run your trial. And with that, we still haven't answered your question, but that's as far as we can go.
It's okay, we're used to it. We don't want to hear.
I think you kind of touched a little bit on this
is the ultimate goal, I'm assuming, is that this would be the drug, the go-to drug. I guess, how does that drug work in conjunction with other medications that our children are already on? Are we going to be stuck with the medicines that we're already on, and this is going to complement that drug, or is the ultimate goal that this is going to be the SCN8 drug?
For me, I guess I can speak for myself and Xenon. You know, the ultimate goal is to get this drug onto the market so that clinicians can determine the best way to use it. And you know, that won't happen overnight because there's established treatment paradigms, and you know that I'm not a clinician, so I can't really comment. But from a Xenon point of view, we want to get this drug to market as quickly as we can. And so, you know, some of that will be determined during clinical development, and some of it will be determined after the drug is on the market.
I don't know if you're going to be able to answer this either, but so let's say we're to the point where we're going to do clinical trials on our children. I'm guessing our children would kind of be one of your go-to people to do that. So I'm envisioning, are they going to have to be off other seizure medications, or as we do this, or are we going to be introducing it and then trying to lower the other ones? I'm just trying to figure out how you can get a true idea. And none of our kids, I wouldn't imagine, are going to be able to take off all their medications and start this one. So just kind of getting an idea, because I know a lot of us are super excited about this. And we're like, yeah, we want to do it, but I don't know if I want you to do it on my kid, because they're kind of doing well right now, and we've seen when it go bad, and so it's like, oh gosh, so we want you to do it, but then sometimes we have this fear of, well, you can do it on someone else's kid, but not mine. So just kind of having an idea of what that might look like, and kind of help us understand, so that way we are maybe more open to and less fearful when you guys come to us wanting us to look at this.
Yeah, my answer to that is that we ask ourselves these questions every day ourselves. That is exactly, and this is really, this is the key point, and this is really to do it carefully and in the right way, that we, and exactly where your question came from in the end, you know, is it as good that this can be the one treatment, or, you know, is it, you know, maybe a great addition to an existing treatment, or is there a different approach? We have to plan that path, and remember, but, you know, first and foremost, you know, we don't want to hurt your kids, and we want to make sure that you know, what we know so far is, you know, from how the drug behaves, you know, preclinically, and how it behaves basically in adult people. So the next steps really it's already quite a quote-unquote risk that we are that you know, we certainly want to come to clinic and come to market as soon as possible, make it available as soon as possible. But at the same time, and I don't want to blame, quote unquote, you know, the regulators and the FDA, and they put limits on. These are really our partners here, and it's actually a great thing to have so many different people at the table looking at the same development program with different eyes, just with the goal, first of all, to keep your kids safe, and second of all, to really potentially have a treatment that might work. But, you know, that's now really, this is where we stand right now, where we are starting to put that roadmap out to answer all these questions. Is you know, co-medication, you know, how do they interact? You know,
that's a big one. I was going to ask next, was the interaction, because so many of us are on drugs that work, but they interact with so many other drugs that we have such a hard time keeping levels and keeping things, and introducing new meds, then it causes another one to go haywire.
Yeah, that's really the goal, to design it in a way that it is, you know, that it can be administered really easily, and that's really the whole goal. I think the modern molecules, as they now are being developed from all companies, they you know, they look at, and I can't speak for the preclinical people, but companies look at these potentials for interactions already way early, way before actually a drug is given. So if there's really a potential for a crazy interaction with one of the more common medication that your kids take, the drug potentially would not even be allowed to go in there. I mean, yes, of course, this is in the early stage we have to prove that. So this is why the first clinical trials are often, we often don't look much at the drug effect yet. Of course, we look at it, but it's really first and foremost how do drug levels behave for certain other drugs? How do safety signals, blood pressures, ability to walk, all that. How does, and it's often very closely monitored and controlled in the very first stages. That will change then in the later stage of clinical development. But remember, until it's proven to be safe and it's effective, we always try to we are all excited about it, but it's also important for us to be as objective as possible because we don't want to move a drug forward in clinical development that we're not sure, but probably it does, but then there's a safety signal that we might not want to see, or so we try to kind of keep that objectivity through that process. But in the end, we can really then convince FDA
and your children. I just wanted to make the point that that's why the survey that we conducted and the information that Dr. Hammer is collecting is so important because we really have then a lot of granularity about the types of medications, the combinations of medications that this patient population is taking. And so as we're thinking about, what drug combinations patients are likely to be on, the number of anti-seizure medicines they're likely to be taking, we really have that information as we kind of plan what the best protocol is going to be to kind of address some of that. So I just wanted to make that point. That's why, that's why we keep asking for this information, is to help us answer some of these questions. Yeah.
And then I'll just add one little preclinical bit to this, and this is these days we screen out any potential very, very early. So for any of the earlier programs that are still in development, we make sure that we understand how they're metabolized, how do they get cleared, would they interact with another drug. And in safety studies, sometimes we look at combinations of drugs just to explore would they have, combined effects, et cetera, et cetera. So I think the industry has really moved to just spend the money preclinically to rule a lot of these out. And then if you happen to find one anyway, you do a lot of studies before you ever get to the patient population, which was already said.
Will the next phase on patient be just US, Canada based, or you're thinking also to enroll patients around Europe? And if yes, how to, you think, to identify hospital centers around Europe?
Our plan is to file, with Neurocrine, what's called an investigational new drug application in the U.S. By the middle of 2020. And so by extension, that means that the trials will start in the U.S. I can't comment further about ex-U.S. or international sites, but certainly the plan is to focus in the U.S. in the first instance.
I can add to that. I mean, since we just came on board so recently, just a few days ago, I don't have a more specific answer for you on that, other than that we are well aware that this is a condition and there's a need that's not only there in the U.S. And we also know that Xenon engaged with multiple experts around the world on that, and we will absolutely build on that. So
I unfortunately can't comment since I don't have any of my clinical colleagues here, so apologies for that.
This is for Xenon. Is your molecule just going up to the neurological, or is it going to affect the cardiac also? because most of our kids have both neurological and cardiac issues with the SCNE and mutation. So I guess my concern is it just going to affect one, or is it going to take care of both?
Sorry, was that Xenon or?
Yes.
Yeah. So XEN 901 is not specifically targeted to the brain. That's where we hope for the therapeutic effect, but it will be found in all tissues in the body. However, it's been designed specifically to inhibit the sodium channel 1.6, which is one of the major excitatory. It is expressed in the excitatory neurons of the brain, and so that's where we expect the major therapeutic effect to be. The sodium channel that's expressed mostly in the heart is a different sodium channel called 1.5, and XEN 901 does not inhibit 1.5 at concentrations that we expect to be therapeutic. So that's a long way of saying we don't anticipate that it will affect 1.5 in the heart. I can't guarantee. That would be the preclinical prediction. That's as far as I can go. It's science, right? I mean, I can't really say for sure.
I mean, from a safety point of view, I mean, all body systems will be monitored in the clinical trials based on,
Does that answer your question?
Kind of.
Does that answer your question as far as we can probably answer it?
I was just wondering if it was protein-based or molecular-based, so it sounds like it's more...
It will inhibit the 1,6 protein.
From a safety perspective, obviously you're monitoring everything from a safety perspective, but what are the types of things that you would expect if there was a safety signal of some sort that you would see with agents like this, that you're going to specifically monitor for, although you're looking at everything? And then from a drug-drug interaction perspective, in an epilepsy study, do you do DDIs with basically all the major epilepsy drugs in the research, in the preclinical work?
So I can say, maybe to the DDI question. So preclinically, we look for how the molecule is metabolized. And by understanding how it is cleared out of the system, you can then say, well, it's mainly cleared by this one enzyme. So let's make sure we understand that how it would interact with molecules that are also mostly cleared by this enzyme, because they might be competing for clearance. So you would do those types of studies early on, but I can't speak to the clinical side, so maybe one of my colleagues wants.
I mean, from the clinical side, I mean, the symptoms that we would be looking for, or the signs or laboratory findings, is the ones that you would otherwise also see with other sodium channel blockers that are on the market and prescribed potentially even to your children. I mean, all the way from more general side effects that we're always concerned about in antiepileptic drugs, such as somnolence, for example. Problems with concentration, even restlessness. But these are not, these more general findings, even down to laboratory findings. EKG is something that will certainly take a close look. While again, back to the cardiac question, that's basically as for this molecule, the hypothesis what preclinical data shows so far is very selective on 1.6, so it should not, affect heart, but that basically also has to be shown, in clinical trials, and not only in healthy adults, but also, in children that are not only affected by disease, but also, have other treatments on board where interactions might pop up that we wouldn't even expect potentially. So so we would monitor that really closely.
Maybe for all of us, can you just walk us through sort of the generic timeline from filing the IND and recognizing you can't give specifics because every drug's slightly different, but just sort of what the sort of regulatory steps are from this point for both companies that were probably the same to get the drug actually to full commercialization and sort of hypothetically how long that takes for a typical drug, recognizing I know they're all very different in terms of the pathways they take.
I really don't have a good answer for that question. And the reason is that this is that there's at the moment and that's we've been thinking about that a lot. How to the goal is to get to the market as soon as possible, right? And we know that, especially from the regulatory agency point of view, there is really an openness to make that path as quick and as fast as possible. In a classic setting, common disease, phase 1, phase 2, 2 phase 3 trials, development program like that can take 7 years, 10 years, whatnot. This is completely clear that in rare indications with a high unmet medical need, the regulatory partners are willing to cut that time really down to really shorter time frames. Is that going to be 6 months? Probably not. Is it going to be 10 years? Probably also not. Where that will end up, it will depend a lot on how the trials are being designed, how the FDA, we will start, and it's already been happening. There's going to be lots of crosstalk between the FDA and ourselves, and probably also the patient community, because in the end, what are good endpoints to prove efficacy of the drug? It's all that. Does the FDA agree on that a particular endpoint is a good endpoint? If not, that doesn't necessarily think that's a bad thing. That just means that we probably need to work together in developing these endpoints to prove the efficacy of the drug. So it's a careful step to be as fast as possible, but also as thorough as possible. We know that the FDA has been willing to kind of completely break up that phase 1, 2, 3 kind of approach in rare diseases, meaning that there's especially in rare epilepsies, even the FDA is saying, as early as possible, we should start looking at efficacy and designing trials in a way that could be proven for efficacy. So that's the middle where we are, to basically design it in a way that it's as straightforward as possible.
So I'm obviously not a clinical trained person, but I think something that also to keep in mind, and I think the community is very aware of, just because you have FDA approval doesn't mean you're going to have payer willingness to pay for the drug. And we're going in, the community is going into more rare indications, smaller patient populations, higher drug prices. Really the onus is on the trial to prove that, yes, there's a statistically significant difference, but it actually makes a difference to the kids and the families. And so that's something that all of us are taking more time to really think about and make sure that we get right.
My question is going to be about potential roadblocks, and you guys kind of answered that in regards to, I think that's one of the things. Obviously, a lot of us, particularly those who don't have great seizure control right now, would love the drug and would say, hey, this is something that needs to be potential. So that was great to hear.
Please forgive me if you guys covered this last night. I was out registering people going in. But, and I have a very rudimentary understanding of everything, but from what I've seen, you've got gain of function, loss of function, and sodium channel blockers, I believe, work best with gain of function. And Praxis said that theirs was kind of like a sodium channel blocker. Do you anticipate that these two medications would be better for the gain of function? Would you anticipate that it would work at all for the loss of function, or are you kind of targeting one side or the other, or you're not sure?
It's not a clinical question. So I will not speak for the Xenon molecule. I will let them speak about how they think about the molecule. For the Praxis molecule, definitely gain of function would be for SCN8, gain of function would be what you would think would be the most likely place for a molecule like ours. That's not to say that SCN8A gain of function is the only place for a molecule, because we do inhibit other isoforms. So Praxis 330 actually, for those of you who don't know, came out of a cardiac arrhythmia program for Gilead. And so and if you go into the literature, there's 20 or so different studies showing that it has anti-arrhythmic, so it maintains normal heart rhythm in various animal models. So there's a lot of cardiac safety data there preclinically. There's also data on NAV 1.4 gain of function. There's data on NAV 1.2 where it's restoring normal proper function in the heart. Interesting, in Dravet, there's some animal data where you would think a sodium channel blocker wouldn't be indicated, but we actually are protected in those mice, probably by blocking NAV 1.6, so SCN8A in the context of loss of SCN1A. So there's the Praxis 330 molecules it's a tool that could be used, or it's when you go to your toolbox and in your garage, it's a tool that could be used in many different instances, but the most likely place initially, as you're trying to prove efficacy, would be gain of function.
Yeah, I would agree that's the same for xenon. It's targeting gain of function mutations in 8A with potentially broader applications, as well as we saw from Marianne Meissler's talk with her ASO work last night, downregulating SCN8A function in Dravet mouse models.
Are there any more questions?
Related to the last question what was asked, like majority of us, we don't know or we're not sure is it loss of function or gain of function. It's like we think this what it is, but nobody ever tried every variant of mutation to put in this.
I'll just add that most of the loss of function SCN8A literature that I have read, they don't tend to have seizures. Am I right? It tends to be intellectual disability, and so I think we have to be careful about how we're determining whether it's a loss of function. The gain of function is relatively easy with some of them. We've done some electrophysiology on this
If
one.
there is a seizure, it's a gain of function.
Not necessarily, but there are a few cases where I think loss of functions, and Chris can add to this, but from the literature that I've been involved in reading, loss of function kids generally don't have seizures. Is that correct? Chris, would you?
That's the way that I understand it, but it is a spectrum, and there are lots of outliers and variations to the rule. In terms of mutations, so you talk about how many mutations have been characterized, and that's something that is definitely a need in this space. Not to get into the hand-waving, not to get into the reasons why we can't do it, but basically this used to be a project that you'd go into the lab, you'd give them a set of 4 mutations, and a year later you would see did they actually make the DNA for those 4 mutations so that they could run the experiment. The actual gain of function, loss of function test may take a month, but it would take a year to make the DNA. We know why that is now. There are labs that have fixed that, they're about to publish on it, and that's timely because what that allows us to do is make tens or hundreds of these mutations, or maybe all of the mutations, put them on now what are the automated patch clamp instruments, and very quickly go through and look at the main properties of the channel. How does the channel activate? How does it inactivate? And then then we can probably do a better job correlating, is it a gain of function, loss of function, or is it mixed? And then how does that correspond to how that mutation presents in the clinic? It's just been really tough, but I'm happy making the mutations. I'm happy to say I think that's now solved and we can move it forward.
It's just a curiosity. My curiosity is not related to the compounds that you are developing. Is it true that the de novo mutation, sorry for my voice, it's true that de novo mutation correlates with a more severe phenotype than an inherited mutation? And if yes, why?
So most mutations in SCN8A are de novo, but there is still a spectrum of severity. So having a de novo mutation doesn't really speak to the degree of the severity of the clinical presentation in that individual. And now I've forgotten the next part. Well, that could be true. So I know within this community there's a number of cases where the parents are mosaic, and so that can be an inherited mutation where a portion of the cells in your body have the mutation, in the parent's body have the mutation, and a portion don't. And so then it depends on, which variant the egg or the sperm carries. So that would be a case of an inherited mutation. Is that more likely to be less severe? Not really, because it just isn't manifesting the disease in the parent because not all of the cells carry that mutation. So it isn't necessarily true that an inherited mutation is has a less severe clinical presentation. Does that help?
Yeah, it's probably the flip. It's probably the severity of the mutation and the presentation, which is driving whether or not it is passed on to offspring. If it's a very, very severe mutation, there's a lower probability that you'll pass that on. If it's a more tolerated mutation, it's more likely to enter the gene pool, so to speak. It's probably the severity that's driving whether it's severe.
I'm sure with it being a small population, it's going to be difficult to do this, but not as an endpoint, but would we have learnings coming out of those studies that would show efficacy specifically to the specific variants, so that we would know, it works better or not as well on certain variants?
We didn't present this data last night, but we have shown in the past cellular data for multiple variants, multiple gain-of-function variants in cell lines that we've constructed. And XEN901 does inhibit the function of all of them that we've tested, except for one. If JP was here, he would know exactly which one, but I don't know which one. But what I do know is the one where it doesn't work is the one that we know that's where the drug binds the protein. So it's perhaps not surprising that the conformation of the protein is changed in the area where 901 is binding. Due to the mutation, the 901 won't work on that specific variant. But we're not far enough ahead in our clinical design to, would we exclude that patient, with patients carrying those variants from a clinical study? I can't say.
I would just say ditto. It's basically the same. Praxi30 works in all mutations that we've tried. There will be mutations where it doesn't work. Well, there will be places on the channel where Praxi30 binds that if a mutation exists there, you're not going to have efficacy. We haven't encountered any of those. Curiously, there is a work from Al George on the cardiac channel where there was a mutation in the prototypic lidocaine binding site, so the local anesthetic binding site, where the cardiac antiarrhythmics did not work when they took that channel out and put it into cells. The things such as quinidine would not correct or would not even inhibit that cardiac mutant. Praxi30 would. So there's something unique about it compared to that binding site. But it's like every other target on a protein. There will be mutations wherever you interact, and unfortunately we won't be able to interact with those.
They just saw those things,
So I think we've come to the end of this session. So just take a moment to thank the folks from Praxis and Xenon for answering some of your questions.
I just wanted to say thank you guys for coming, because it's so neat to sit in the back of the room and hear everyone ask you questions. I can pick up the phone and call Allison, and I bug her with questions all the time. I bug folks at Praxis all the time with questions, and I just am so excited to get to see you guys bring your questions directly to them and hear them answered by real people, not mediated through me on Facebook. So thank you for, of course, I'm not surprised at all that you were filled with questions and willing to share them. So thank you guys for participating, because that makes a very big difference. And thank you guys for coming. It's fantastic to have you. So I think we're going to do a quick 5-minute break. We'll get Tracy set up for her